Okay, so we left off with this lovely slide here, one of my favorite memes here, where we were talking about how we can classify neurons both structurally and functionally. And so we're going to first talk about how we can classify them structurally. And so if you look, we have multipolar neurons, bipolar neurons, and unipolar neurons. Now, I'm going to start with unipolar neurons because it's easiest to see why they're called unipolar. If you look here, you have this kind of one process, uni, right? Uni means one. This one process coming off of the cell body. And it actually forms this kind of T-like process into two axons. So it branches basically here into two axons. And so if you look here, then this end, of course, is going to end up in your axon terminals and the synaptic end bulbs. And then here, so this is going to be the end that's going to be putting out neurotransmitter, right? And passing along the message. And this end here is the dendrites, has the dendrites that are going to be receiving the message. So again, it's called unipolar because, again, it has this one process coming off of the cell body. And you're going to see that these are very commonly going to be found in your peripheral nervous system. And they're going to be mostly sensory neurons. So you're going to see we're going to classify these functionally in just a minute. And I told you there's going to be crossover. So again, most of your sensory neurons are in this category. Most of the sensory neurons are unipolar structural neurons. So that'll kind of help with a little bit of crossover. And so again, oftentimes these are associated with sensory receptors. And again, so they're taking that information from the sensory receptors, again, back to the central nervous system. And usually, again, the proximal processes actually enter the central nervous system as, again, they're going to pass on that message from those sensory receptors to the central nervous system. So just a kind of crossover. I know I'm crossing over kind of the structural and functional, but it's hard to, again, like this, right? Like it's hard to talk about structure without talking about function. They go hand in hand so much. Okay, so the second structural classification is going to be bipolar. And this is, again, pretty easy to see because you're going to have now bi means two processes coming off of that cell body or that soma. So if you look here, you're going to have one axon coming off in this direction and one dendrite coming off in this direction. So these are a little bit more rare. You don't see them. They're not as common in especially they're not as common as unipolar, and they're definitely not as common as multipolar. So you're going to see these in kind of like special senses, especially when we talk about our kind of special sensory receptors. So we're going to find them in things like the retina of the eye and the olfactory mucosa in the nose. These are really going to be associated more with our special senses. And then the last one here is going to be your multipolar neurons. And so again, it's easy to see that this is multipolar because now instead of having one or two processes coming off of the soma or cell body, you have more than two. So you have many, right? So 3 or more processes are coming off of this cell body. So here you can see in this multipolar neuron, you have lots of dendrite processes coming off, right? So not just one, but many dendrites coming off, okay? And you're going to see it also has, again, a single axon coming off. So one axon and then many dendrites. And this is going to be the most common of all of your structural neurons that you're going to see in the human body, the one that you're going to find in the greatest abundance. And it's going to be your major neuron type in the central nervous system. And these also make up many of what we call your motor neurons as well. Okay. So those are the different structural classifications: multipolar, bipolar, and unipolar. Pretty easy, right? Okay, so now we're going to talk, here's just a nice little kind of review chart of looking at these different structural classifications: multipolar, bipolar, and unipolar neurons here. And again, just another chart kind of showing you the structural variations. So it's just showing you the like different variations that you can see upon a multipolar neuron, what the different variations are on a bipolar neuron and a unipolar. But again, all of these are going to still fit those structural classification requirements. Now, what's interesting, in unipolar neurons is that you're going to see that these cell bodies, oftentimes they're going to be found in what we call a ganglion. And we'll talk about this a little bit later here at the end of this lecture. But basically, you'll have lots of unipolar neurons like running alongside each other. And then all their cell bodies will kind of be hanging out in this one area that we typically call a root ganglion. Okay, so again, I'll talk about this a little bit later at the end, but this is what kind of forms our ganglia, is this kind of grouping of cell bodies in our peripheral nervous system, especially of the cell bodies of a bunch of like sensory neurons. So again, just to kind of introduce that. Now we have the structural classification, then we have the functional classification. So our functional classification is going to describe kind of the nerve impulses and relate what direction the nerve impulses are traveling relative to the central nervous system. So we have our sensory neurons, which are of course going to be transmitting impulses towards, right, the central nervous system, towards the brain and the spinal cord. And again, like we talked about, almost all of these are going to be unipolar. So again, there's the nice crossover between structural and function. And then we talked about just a minute ago that the cell bodies are going to be located in what we call these ganglia, these kind of groupings of the cell bodies in the peripheral nervous system. And then we have our motor neurons, which are going to be carrying impulses from the central nervous system to effectors, to things like, again, our organs, or to our glands, or to our muscles, okay? And these are going to be multipolar neurons, okay? So lots of and this is where we talked about lots of multipolar neurons in the body, because we talked about also the multipolar neurons are going to be in the central nervous system, which we're going to talk about in just a minute. So again, you can see the majority of our neurons are going to be multipolar neurons in the body. And most of the cell bodies here are going to be located actually in the central nervous system. Because again, remember that they're taking the message out. So the dendrites are going to be in the central nervous system, picking up, again, that information from those integrative neurons in the central nervous system that are telling them what to do. And then they're going to carry that message down their axon out through the peripheral nervous system to a muscle or to a gland. Okay, so that's why most of their cell bodies are actually going to be located in the central nervous system. Again, except for some of these autonomic neurons that we'll kind of get to later. And then we have the third functional classification of neurons, which is going to be our interneurons, or our integrative neurons, or our association neurons. I know they have lots of different names here. And so these interneurons, I like interneuron because inter means between, and technically your interneurons are between things. They're usually between sensory, right? They're usually between sensory and motor neurons. So I like the term interneuron because it kind of tells you where you typically find them. But again, they're also called association neurons, integrative neurons. You'll hear them called all three. Your book actually even goes back and forth. And again, they're inter because they lie between motor and sensory neurons. And they're taking signals, again, they're kind of shuttling signals through the central nervous system. This is where, again, kind of the decision-making process is happening, right? As sensory is coming in and you're deciding what motor output is going to be going out. And so this is 99% of the body's neurons are actually interneurons, okay? So majority of the body's neurons are interneurons. So again, this is just a kind of that classification talking about the now more functional classification of these interneurons, and talking about, again, where you might find some of these neurons, okay? And again, we didn't talk about some of the bipolar neurons that make up your sensory neurons, but your bipolar neurons are going to be, again, these special senses, sensory neurons, taking in information from the eyes for sight and the, nose for olfactory. So again, those are going to fit into, it's easy to picture that those are going to fit into the sensory neuron category as well. Okay. So hopefully that kind of wraps all that up for you in terms of the classification of neurons. Again, as always, please let me know if you have any questions on those. And then now we're going to get into these neuroglial cells. We always talk about the neurons, but again, the neurons can't do their job without the neuroglial cells. So I love this little, cartoon here. And the glial cell's like, hey, why doesn't anyone talk about me? The neuron's like, oh, I'm doing all the major work. But the neurons, again, can't exist without these neuroglial cells because the neurons are so busy doing their job of taking in messages and transmitting messages in these forms of these electrical signals. They're so busy with that they don't have time to really take care of themselves, feed themselves, do some housekeeping, clean up the area around them so that they can continue to do this job. So again, these neuroglial cells are really, really important. Half of the volume of the central nervous system is made up of these neuroglial cells, as you can kind of see around here. And they're 50 times more numerous. Again, easy to picture. They're, oftentimes much smaller, but much more numerous. And these cells can divide pretty rapidly. Unfortunately, because of that, this is where we typically see neoplastic growth. So this is where we see tumors. And so this is what I actually did my graduate research on, was I did my graduate research with gliomas, and specifically stage 4 gliomas. So again, unfortunately, because these cells can divide rapidly, they also are the cells that form tumors. And again, that rapid mitosis can allow for rapid growth of these tumors. So that's just something to keep in mind as we talk about some of the clinical aspects of this. When you're talking about brain tumors, you're usually talking, again, about these tumors that are in the different types of glioma, neuroglial cells. And we'll talk about, you have these different types of neuroglial cells. So some of you may have heard of like astrocytomas. That's because it's going to be, a tumor growth with astrocytes. So again, just kind of, if you've heard of some of these different types of tumors, then these might actually help you kind of make sense, because some of you may know someone that's had one of these types of brain tumors. Okay, so when we talk about these neuroglial cells, we're going to break the cells up into the neuroglial cells that we're going to find in the central nervous system, right, around the brain and the spinal cord neurons. And then we're going to talk about the neuroglial cells that we're going to find in the peripheral nervous system, okay? So again, these are going to be surrounding those neurons that we're going to find in the nerves, the sensory nerves, as well as the motor nerves, okay? And that's going to be in your peripheral nervous system. And anytime I should have explained that, anytime I use CNS, that's central nervous system, and PNS is peripheral nervous system. So again, your central nervous system is your brain and your spinal cord, and your peripheral nervous system is going to be all of the nerves that are coming to and going to the brain and spinal cord, okay? So when we talk about the central nervous system, we have 4 major types of neuroglial cells that kind of take care of our neurons there. And that's going to be your astrocytes, your oligodendrocytes, your microglial cells, and your ependymal cells. Ependymal cells, we'll get into all those in just a minute here. And then we have 2 types in the peripheral nervous system. We have your Schwann cells and your satellite cells. So let's get to it. The first group of cells we're going to talk about are these 4 cell types in the central nervous system. And so the first one is going to be your astrocytes. So these guys are the most abundant, they're the most versatile, and they kind of cover the most jobs that need to be taken care of in the central nervous system. They're actually going to have these extensions that cling to the neurons, and the synaptic endings, and even the capillaries. So you can see a lot of times they'll have these extensions that go from the neuron to the blood capillaries. So this is in the central nervous system. So again, in the brain, in the spinal cord. So it's pretty easy to kind of figure out what one of their major jobs is. If again, they're attached to a blood capillary and that same astrocyte is then attached to a neuron, it's pretty easy to figure out that these guys are going to help transport nutrients and oxygen, things like that, to this neuron, from the capillary and to the neuron. And so you may say, well, why don't the neurons just directly take in those nutrients from the blood capillary themselves? And again, one of the reasons is that neurons are just simply too busy, right? But also another reason is to help protect your neurons from harmful substances. So these astrocytes kind of act as a filter, a filtering cell, kind of bringing in the things that are good for the neurons and then trying to isolate the things that are potentially harmful from the blood, from the neurons. So again, it's kind of this layer of protection. So these astrocytes, again, are going to play a role in making exchanges between the capillaries and the neurons. And they're one of the cells that make up part of that blood-brain barrier, okay? So again, they play a role in making exchanges between the capillaries and the neurons. They're going to, again, bring in nutrients to those neurons, but they're going to help make sure they filter out. So here's an example of that, an astrocyte here doing that, but they're going to make sure they filter out, again, some of the potentially harmful stuff. So pretty cool, right? And again, so obviously they're going to be transferring nutrients from the capillaries to the neurons. So again, these neurons are just far too busy to feed themselves. So they actually have these special astrocytes to feed them. Now, one of the reasons that they're called astrocytes is if you think astro star-like, they look kind of star-like because they have all these extensions coming off in all different directions. So that, again, they can go from a capillary to a neuron and to another neuron. These cells are also going to provide a lot of support for the neurons, kind of brace the neurons and help to kind of hold them in place. So you can kind of picture them doing that with all of these extensions as well, helping to really brace neurons and hold them in place. And then you can see they're also going to help regulate the growth of neurons, the migration of neurons, meaning as these young neurons are forming, where those neurons are going to form and what connections they're going to make. Astrocytes will kind of help with that migration and help to hold them in place in the right place as these new neurons develop. And then also, again, they're going to be involved with helping hold these neurons in place as they make these connections with other neurons. And then they're also going to help kind of control some of the chemical environment around the neurons. So they're going to help to kind of regulate the ion transport, okay? Again, making sure that the right ions are surrounding these neurons. They're also going to help metabolize any excess neurotransmitter. So pick up any excess neurotransmitter, and then sometimes they'll take that excess neurotransmitter and then give it back to the neuron. So then again, that neuron can recycle it. So again, those are all major functions of the astrocytes. As you can see, the astrocytes really do a lot. They're busy cells. And so again, here is your, oh, I don't think I have, do I have the other picture? Yeah, here's your astrocyte. I like this picture of the astrocyte because it really shows, again, why you call it an astrocyte. It looks very star-like. Okay. And then our next group of cells, so our second cell that we're going to find inside the central nervous system is going to be the microglial cells. Okay, and these cells are our defensive cells. So you're going to see they're going to be kind of oval in shape. They're typically a smaller neuroglial cell, and then they're going to have lots of these kind of like these like kind of thorny, they call them thorny extensions because they're really branched. Lots of these branched extensions coming off, okay? They look kind of like thorny. So again, they're these kind of small, ovoid, oval in shape These cells or these thorny processes, they're typically usually found near blood vessels. So you a lot of times find these microglial cells here near blood vessels. And that's because one of the things that they're going to do, one of their major functions, again, is to provide kind of this layer of protection. And also, they're going to kind of be your cleaner cells. So they're kind of your house cleaners of the central nervous system. So these guys will typically oftentimes migrate towards an injured neuron, and they'll help transform. They'll transform themselves into these cells that can engulf things, phagocytize. So I don't know how much I've used this word in my previous lectures, but phagocytize means to engulf. Basically, it's like cell eating. And so when a microglial cell comes upon, maybe a potential microorganism like a bacterial cell in here, it's going to engulf that bacterial cell and break it down, okay? And to help protect, again, those neurons from infection. So that's one of the major functions of these microglial cells, is to kind of provide this immune function to protect those neurons, those central nervous system neurons, from bacteria and viruses. Okay? Now, they're also going to clean up any cell debris as well. So, for example, again, if you've got some, dead cells here that have kind of broken apart and are kind of hanging out here in the central nervous system, they're going to basically do cleanup there. So they're going to clean up all that debris. So that's kind of their major function. And as you can see, it makes sense that these would be derived from cells that are going to eventually give rise to these immune cells that are outside of the central nervous system, that are going to be engulfing bacteria and viruses and things like that. So those are things like your macrophages and your monocytes. I know we probably haven't really talked about those yet, but you will in 202. So anyway, so your microglial cells, again, are these cleaner cells, and they're really providing a big immune role here as well, cleaning up bacteria and viruses and things like that. Now, your ependymal cells are going to be these specialized cells that are going to help kind of contribute to that barrier between the blood and the brain, especially forming what we call the blood/cerebrospinal fluid barrier. So now, cerebrospinal fluid, we haven't really talked about cerebrospinal fluid, but cerebrospinal fluid is going to be the stuff that bathes the brain and the spinal cord. It's going to be the specialized fluid. It will be clear, and it's going to provide the kind of perfect balance for, again, the neurons in the brain and the spinal cord. So it's going to provide like the right pH, the right ion concentration. It should be clear. They're going to make sure that these cells are going to make sure that no bacteria are going to be entering again, or as they make the cerebrospinal fluid, that none of the bacteria from the blood is going to be used to make that cerebrospinal fluid. So again, in a healthy individual, your cerebrospinal fluid, if you've ever seen someone that's had like a spinal tap done, their cerebrospinal fluid should be very clear. Unless, again, if it's cloudy, that's usually an indication that they have some type of infection. And that's usually, again, a more kind of serious thing that is going to be treated very quickly, because obviously you can see you don't want to have bacterial cells running around your cerebrospinal fluid, around your spinal cord, making their way into neurons in the central nervous system. It's not a good thing. So again, these ependymal cells are going to line certain cavities of the brain and the spinal column. And we're going to talk about these cavities coming up here, especially your ventricles of the brain. And then we'll talk about in the spinal column where you're going to find the cerebrospinal fluid. And again, these cells are going to make that cerebrospinal fluid. So your ependymal cells you can thank your ependymal cells for making that cerebrospinal fluid. And they're going to do it you're going to have lots of these cells in a place called the choroid plexus. Again, I know you don't know where this is, but I'm going to be introducing this when we get to the brain. So I just kind of want to mention it now. So right now, you just have to know that the ependymal cells make the cerebrospinal fluid. But we're going to talk about, again, when we get to the brain, that they're actually concentrated in this area called the choroid plexus in the brain. And we'll talk about that. Again, these guys may be ciliated. So if you look here, here's all your ependymal cells right here. And they're forming this, again, this nice blood cerebrospinal fluid barrier. And you can see that they're going to have these little cilia coming off here. And those cilia are, again, going to beat and circulate the cerebrospinal fluid. So really important for that. So here you can see, again, those cilia circulating that cerebrospinal fluid. So again, these ependymal cells are filtering, and they're typically, again, going to be associated with blood vessels. And what they're going to do is they're going to filter out the fluid from the blood and use that to make the cerebrospinal fluid. Okay? So that's basically their main job. The third category of cells that we're going to talk about are going to be your oligodendrocytes. And these oligodendrocytes are pretty cool cells in that they are going to create what's called the myelin sheath that goes around your axons. Now, these guys are going to be located only in the central nervous system. You're going to see that these are going to be very different from your Schwann cells, because you may be like, well, wait a second, I thought Schwann cells were the cells that make the myelin sheath. Okay? Schwann cells are going to make the myelin sheath in the peripheral nervous system. But they're going to be different. They're going to look different. And the way that they wrap around and form that myelin sheath will be different than the oligodendrocytes in the central nervous system. So they're actually two different types of cells, and it's really important that you differentiate them. So again, oligodendrocytes are only in the central nervous system. Schwann cells only in the peripheral nervous system. Okay? Make sure, hint, hint, that you know that. So oligodendrocytes are going to be these cells. Here's a perfect example. They look kind of like an octopus, right? And they're going to be these really branched cells. They're going to have these processes that come off of them and wrap around the central nervous system axons, okay? Forming, again, this nice insulated layer called the myelin sheath. And each of these oligodendrocytes will actually wrap around more than, that's my greater than sign, more than one axon. So you can see here, again, it's wrapping around, and they form these little kind of segments, right, of the myelin sheath. So here, not only is it wrapping around this axon for this neuron, but it's also wrapping around this axon for this neuron, and then also wrapping around farther up here to make a different segment of that myelin sheath, okay? So that's your oligodendrocytes. And you're going to see that this is going to be very different from your Schwann cells, okay? So very, very different in just a minute. So again, that myelin sheath, what is this myelin sheath that we talk about here? Why do these oligodendrocytes even do this? And so the reason is that this myelin sheath is going to act like the insulation to your wires, okay, to your electrical wires. And so the myelin sheath is actually white, and that's because it's fatty. So it's this protein lipid substance. And again, like I said, it's a fatty substance. It's got a lot of lipid in it. And that is really going to help to provide insulation for your myelin sheath, for your neurons here. And that's because, again, remember that we are going to be transmitting electrical impulses down this axon. So again, just like a wire, we're going to want some insulation to allow that electrical impulse to be efficient and fast, okay? So again, we're going to see our myelin sheath produced in Schwann cells and oligodendrocytes. Even though they're going to be produced by different cells, they're going to look similar. And if you look here, what you're going to end up seeing is that as this extension comes off of this oligodendrocyte here and it wraps around that axon, it's mostly going to be that phospholipid bilayer that you're going to see, right? And so basically what you're doing is you're taking that phospholipid bilayer and you're like wrapping it around like a jelly roll many different times. And so that phospholipid bilayer is really going to help prevent things that are large and charged from moving across that axon cell membrane, what we call the axolemma, right? And so again, this myelin sheath is going to surround the axon of most neurons, not all, but most. And that sheath is going to, again, protect the axon from damage. And we'll talk about this. This is really important because there are some diseases like multiple sclerosis that attack these cells that are making the myelin sheath, and then they leave the axon of the neuron vulnerable to be damaged. Okay, so protection of that axon is one of the really important functions of the myelin sheath. So, but not only do they protect it, but again, they insulate it electrically, because again, we're going to be sending electrical impulses down there, just like, again, the electrical impulses that are sent in your electrical wires. And so this is going to allow that myelin sheath will allow the electrical or nerve impulses to be conducted at a faster rate, okay, because they're insulated. Now, we do have some fibers that are non-myelinated, so they don't contain this myelin sheath, and they do conduct impulses more slowly. But again, we'll talk about reasons why we have this, and again, you're seeing this mostly in the central nervous system, especially as we get into the brain. Again, this is just kind of showing you this oligodendrocyte and all these extensions that come off, and how, again, it forms this kind of jelly roll wrapping called the myelin sheath around multiple axons. So again, that's in the central nervous system. So that's sorry, that was my fourth cell, okay? So again, for the central nervous system, sorry, I meant to go this way. For the central nervous system, we have, for our cells, microglial cells, okay, our kind of cleaner cells, our ependymal cells, the ones that are making the cerebral spinal fluid, or astrocytes, which have lots of functions, including, again, feeding the neurons and supporting them and making sure, again, that they kind of filter things for those neurons. And then we have our oligodendrocytes, which, again, are forming the myelin sheath around the axons in the central nervous system. So those are our four major central nervous system neuroglial cells. Now we have just two major neuroglial cells in the peripheral nervous system, okay? So again, in our sensory neurons and our motor neurons. And so these two different types of cells are called satellite cells and Schwann cells, okay? So as you can see, our satellite cells here are going to surround the actual cell body, or the soma. And then our Schwann cells are going to surround the axons in the peripheral nervous system. Okay, so let's talk about these. Let's talk about satellite cells first. These are these kind of flat pancake-like cells that are surrounding, again, your soma or your cell body of your neuron. And they're especially going to do this in these peripheral ganglia. So remember these, what we called unipolar, remember? Because it's, again, just got this one extension coming off of the cell body. This is especially common in these unipolar neurons where this cell body is just kind of hanging out there. So we have these nice satellite cells to provide some protection. They're very similar. They have kind of a similar function to the astrocyte, to the central nervous system. So again, these guys are going to really help to filter out. They're going to help to transport nutrients to the cell body. They're going to help to kind of filter what's coming into the cell body. They're also going to help kind of provide some structural support as all of these cell bodies are from kind of multiple neurons going to be grouped together in this ganglia. So they're really going to help support the neurons in the peripheral nervous system ganglia, what's going to be this grouping of all of these different cell bodies from these unipolar neurons. And again, remember, these unipolar neurons are going to be primarily sensory neurons. So mostly your sensory neurons. And so that's kind of, you can see this, this is kind of a nice image of cross-section through the unipolar soma here, the unipolar body here. And then you can see some of these satellite cells around the periphery, kind of, again, forming like a little barrier around the periphery of that cell body. And you can see that here in this nice cross-section as well. Okay, so those are your satellite cells. And then you have your Schwann cells, which are going to be your cells in the peripheral nervous system that are going to surround the axons and make your myelin sheath. Again, it's a different cell. You're going to see it looks very different because you're just going to have each cell is going to be dedicated to just one axon, okay? So it's not branched like you saw your oligodendrocytes were in the central nervous system. So again, these cells are going to encircle your axons only in the peripheral nervous system, okay? And they're going to surround basically your motor and your sensory neuron axons. So again, they're very similar to oligodendrocytes. They're going to wrap around, forming this myelin sheath. But instead of having extensions, the whole cell itself is going to just wrap around. And so it's a little bit different because remember the oligodendrocytes had their kind of the cytoplasm in the cell body basically sticking out and then extensions going off and wrapping around the axon. But here the entire cell is. And so this whole Schwann cell is going to wrap itself around and then it's going to put out these extensions that are going to form, again, the same jelly roll. But the difference is that its cytoplasm and its nucleus are going to kind of get pushed to the outside of this jelly roll, forming this outside segment. And in lab, we call this outer layer here of the Schwann cell that has the cytoplasm and the nucleus. We call this the neurolemma. So just kind of as a reminder for lab. And so again, but you're going to see it's going to form that same jelly roll with, again, this kind of nice phospholipid fatty layer of myelin. That's going to insulate that axon. Okay, so very similar. Now here you can see in what we call unmyelinated axons, you do have in the peripheral nervous system Schwann cells that will surround unmyelinated axons. And you may say, well, how is it unmyelinated? Because the Schwann cell is still around here. But it's not forming that jelly roll wrapping. So this Schwann cell here is actually really kind of just like holding these axons together and kind of keeping them nice and organized. But it's not wrapping around and forming this really thick myelin sheath where you've got layers upon layers, right, of that phospholipid bilayer. So here, again, you don't have all those layers. You don't have a true myelin sheath, okay? So those are called unmyelinated axons. And so one of the things in the peripheral nervous system, especially again, because you're outside of the protection of the central nervous system, in the peripheral nervous system, your peripheral nerve fibers are much more likely to be damaged. And so that's one of the reasons why we have these different types of cells forming the myelin sheath, is because they're really vital to the regeneration of these peripheral nerve fibers. So we really need to have these different cells that form the myelin sheath. And I'll talk about that in just a minute, how that process happens. So again, this is just kind of showing you, again, your Schwann cell, how it wraps around, shoots these extensions of that phospholipid bilayer membrane to wrap around like a jelly roll around the axon, forming that, really the myelin sheath. And then this outer layer that we call the neurolemma is going to contain the cytoplasm and the nucleus of the Schwann cell. It kind of gets pushed to that outer layer. So when we talk about, again, myelination in the peripheral nervous system, we see the same jelly, that wrapping around the axon in this jelly roll fashion of this myelin sheath. But again, one cell is going to form just one segment of the myelin sheath. This, again, hint, hint, is very different from the oligodendrocytes of the central nervous system that are shooting off extensions, and one cell is going to actually form many segments of the myelin sheath on an axon, and it can be on multiple axons as well, okay? So very different, right? And then that outer column we already talked about, it's still called the neurolemma. It was kind of an older term, and we've gone back and forth, but, that should be with an O, neurolemma. It's still called a neurolemma by most individuals. So we're still using that term in lab. And Again, it's that bulge of the outer layer that is going to contain, again, all of the cytoplasm and the nucleus. Okay? So as the tube grows, it's really going to help guide axons that are repairing themselves. So as these Schwann cells kind of grow and wrap around themselves, this will really help guide the growing axons in the peripheral nervous system when damage occurs. So I'll kind of show you how this happens. And so keep in mind that plasma membranes, again, when you're talking about the wrapping around to form that myelin sheath of these plasma membranes, right, the phospholipid bilayers, and you've got many layers, remember that they have less protein than, these are going to have less protein than even some of your normal cell membranes. And they're going to have no channels or carriers, so they're going to make these really good electrical insulators. And so that's kind of one of the big key things to remember about the plasma membranes of the Schwann cells. So again, this is just how it forms, really easy on how the Schwann cells form this jelly roll. Basically, what they do is the Schwann cell, as it grows, it will start to wrap around and then kind of push extensions of the plasma membrane underneath itself. And then as it does, the cell just is going to kind of rotate around the axon, wrapping its plasma membrane kind of loosely around it in these successive layers until it eventually again forms this nice tight jelly roll of plasma membrane around that axon. And this jelly roll portion of the plasma membrane extension is called the myelin sheath, okay? So that's what's providing the main insulation. And you can see that this is happening here. And again, you can see that neurolemma of the cytoplasm and the nucleus that are pushed to the outside, that neurolemma layer there. And then again, this is just showing you what the unmyelinated axons look like, where there's no jelly roll. Okay, so again, here in this case, the Schwann cell is just kind of helping hold axons from different neurons together, keeping them organized. Okay, and so when, again, we talk about the myelination, as you can see, we're going to form, when Schwann cells form their myelin sheath around, they don't cover the entire axon, right? So they'll do it in segments. And this is really important because this is going to leave these little sections here that are unmyelinated. And this is really, really important for nerve conduction. So you have to know these little sections here. Sometimes for some reason students don't think that this is important, and it is actually really important to know this. So these are called the nodes of Ranvier, okay, or nodes of Ranvier. Ranvier is the French, because it was a French scientist that discovered it. So these nodes of Ranvier are really, really important. These little segments in between the myelination, they're basically little gaps, okay, between these adjacent Schwann cells. So make sure you know these, because these are going to be important in our next lecture. So these are called the nodes of Ranvier, or nodes of Ranvier. In the United States, we kind of pronounce it nodes of Ranvier, but if you go in Europe, it's nodes of Ranvier. Non-myelinated fibers, again, we already kind of talked about those. They'll have the Schwann cells, but no coiling. So, you might have one cell that makes around like 15 different axons from different neurons, but no myelin sheath. This is just kind of the structure of the motor neuron to kind of go back out and show you again. This is what this is going to look like with all of these Schwann cells kind of forming these segments. And then here's the little spaces, the gaps in between them, called the nodes of Ranvier. Okay, or myelin sheath gap is the new name, for the hip kids. For all you hip kids out there, we call them the myelin sheath gap. I'll use both terms. I'll try to make sure I use both terms on the test, but you will hear me use node of Ranvier because it's just what I grew up with. So, and I always like to give credit to the scientists that discovered it because, right? I mean, it was pretty cool that they discovered it. So anyway, this just goes through myelinated fibers versus unmyelinated fibers. Myelinated fibers are going to appear white, and that's because of all that fat in them. So basically all the lipids from the phospholipid bilayer, all those lipids, those excess lipids are going to make it look white in appearance. So again, that jelly roll myelin is going to act as a great insulator, and that is really going to help speed up conduction of the nerve impulses in your myelinated fibers. Your unmyelinated fibers, again, are going to have kind of a slower nerve impulse. They're usually typically smaller in diameter, and usually you'll find these again in the central nerve, some parts of the central nervous system. And again, they're only surrounded by that outer neurolemma. Sorry, that's supposed to be a neurolemma, but no myelin sheath wrapping, okay? And we'll talk about why that is coming up here. Myelin sheaths in the central nervous system. Again, I'm doing this as a comparison to your Schwann cells. So remember that, as a reminder, your oligodendrocytes in the central nervous system are going to be forming the myelin sheath in the central nervous system. They are going to, like your Schwann cells, have gaps between their segments as they, again, shoot off these extensions. So we still will have the nodes of Ranvier. They're still going to be present in the central nervous system as well. But there's no neurolemma, okay? There's no outer color because remember that these oligodendrocytes are just shooting off extensions. So again, I'm just going to kind of go back here just to remind you of the major differences here. So remember that these oligodendrocytes are going to look like this. Oops, sorry, that wasn't what I wanted. Your oligodendrocytes, oh, no, it's not here too. I guess this is our best oligodendrocyte. Our oligodendrocytes are going to look like this, okay? So there's no neurolemma because the soma of this cell, so basically, again, the nucleus and all the cytoplasm are going to be over here, and then they're just going to shoot off extensions to form, again, the myelin sheath, okay? So there's no neurolemma on these guys. So again, really a big difference, right, from in comparison to your Schwann cells. And then also, again, keep in mind that each cell here can wrap up multiple axons. So oligodendrocytes shoot off these extensions, and they can wrap up to about 60 axons with all these different extensions, okay, making different segments of the myelin sheath. And so that's kind of the basic. And then you're going to see here, when you're talking about in the brain, gray matter, and we'll get to this more when we get to the brain, but I just want to mention it now because we're talking about myelination. The white matter of the brain is going to be where you have the myelinated axons, okay, inside the brain. And the gray matter is going to be where you have nerve cell bodies, you're going to have dendrites, you have axon terminals. Sometimes you have unmyelinated, bundles of unmyelinated axons. So that's where you're going to find unmyelinated axons, is in the gray matter here. So here's gray matter. And your neuroglial cells, okay? So again, and then here, the white matter is going to consist, again, mostly of just myelinated axons. So that's why it's called white matter. So kind of cool. Again, even when you see that here, you know, your axons are going to be here for these myelinated axons are going to be here, but, you know, the cell bodies and dendrites and all that are actually going to be in here in the gray matter, okay? So again, that's why white matter is white. It's because of the myelin. Just a couple more definitions, and then we'll stop here, and then the next lecture we'll get into kind of actually how finally neurons pass on their electrical impulses. But just a couple of definitions. We have the term ganglion, which is just going to be a cluster of neuron cell bodies in the peripheral nervous system. So again, this is going to be a cluster of cell bodies from unipolar neurons, okay? On the peripheral of these sensory unipolar neurons. And so we call that a ganglion, where we have a group of those. And then this is confusing. I didn't do this. Please, again, don't hate the messenger here. But we use the term nucleus at a macro level, so not inside the cell, but a macro level, to talk about a cluster of neuron cell bodies in the central nervous system. So these are important terms to get used to because we're going to talk about these throughout the next couple chapters. So a cluster of cell bodies in the peripheral nervous system is called a ganglion. A cluster of cell bodies in the central nervous system is called a nucleus. Again, So it's confusing because there's a nucleus inside the cell. This is talking about a different type of nucleus, and you can tell by the context that we use it in. This is talking about, again, a group of cell bodies in the central nervous system, and they're going to serve certain functions when we get to the brain. So we'll talk about that. Then also, we classify bundles of axons that are in the peripheral nervous system and the central nervous system differently. A bundle of axons in the peripheral nervous system is called a nerve. A bundle of axons in the central nervous system, the brain, and the spinal cord is called a tract. So in the central nervous system, in the brain, and in the spinal cord, we don't have nerves. We have tracts. And then in the peripheral nervous system, we don't have tracts. We have nerves as our bundles of axons. And then again, the white matter is going to be your myelinated axons. The gray matter, as we talked about, is going to be cell bodies, dendrites, unmyelinated axons, and neuroglia. Okay, so we're going to go ahead and stop here. If you have any questions, please let me know, and then we will jump right into how we actually transmit messages down these neurons in the next lecture.